Okay, so I just want to take a moment to recap the Microscope Lab and everything we did last week. So you want to make sure you have your two handouts out, which would include the Microscope Lab handout that we started. It has most of this info, in addition to the other part that has the cheek cells and onion cells. So on the name slide, what you should have noticed is that when you moved the slide towards your right hand, the words on the slide actually move towards the left. So the microscope works opposite of how you see it. And also, if you were able to get the little E in the entire field of view, you should have noticed that it was flipped upside down from how it was on the slide. So a specimen that is right side up and facing right on the microscope slide will appear upside down and facing left when viewed through a microscope. So just a little tidbit about your compound light microscope, that it changes how you see it, pretty much opposite and upside down. That was the point of the name slide. And then when you look at, that's not the one I want. Okay, salt versus sugar. So for salt versus sugar, this is sort of what it should have looked like under a microscope. You looked at it with just your eyes, then you looked at it under the microscope. And salt crystals, you can see here, are pretty much in cubic form, whereas sugar crystals are not cubes, but oblong and slanted on the end. So that's one of the biggest differences between salt and sugar. For the next, I think it was part 4, for the prepared slide. So you looked at a lot of different slides, and really the whole point of looking at all those prepared slides was just to get used to using your microscope and following the proper procedure. Making sure you have a clear image. And I know it was hard to draw, different things, but in general, I just wanted to talk about a couple of them. The slide that you looked at that was bone was not dyed a certain color, so it didn't look as nice as this one did. And that would obviously be made of animal cells versus an elder stem, which was one of your slides, are plant cells. And when you're looking at them on a microscopic level, you should see some differences. And some of the differences are going to be cell size and shape. Differences between plant and animal cells. Plant cells are usually larger. So this whole thing right here that I am sort of highlighting with my cursor, that is one plant cell. And they are usually rectangular. And we know why they're rectangular, right? Because of their cell wall. Versus an animal cell here, this is frog blood cells at 400x and stained purple, are smaller and circular. So those are some big differences between plant and animal cells under microscope. And then when we're looking at cheek cells, one thing I wanted you to note with the cheek cells, a lot of you could not find a really great cheek cell sample. But if you did, this is kind of what it looked like here. And this is a really great sample. I think this is probably under 400x of cheek cells. And if I were to show you a diagram of this, then the darker part inside every single cell that we saw is the nucleus. And the reason why it stains so dark is actually because of the chromatin or the DNA. And then you might see little structures within the cell itself. The space is the cytoplasm. The outside of the cell is the cell membrane. And some of the larger organelles that could have been picked out would be the mitochondrion, which is really cool. So you can see some cell organelles while looking under the microscope. This is for a plant cell here. And in a plant cell, if this is one cell that we're kind of zooming in on, it's hard to see the outline, but in the center where you don't see anything else, that is the large central vacuole. The green parts in a plant cell would be the chloroplast, and outside you can see a rigid cell wall. Onion cell and water. This didn't work out for a lot of you all, what I was wanting you to see, but I was wanting you to look at really the outside of each cell. So you can see the cell wall is intact here, just in water. And this was sort of an introduction to cell transport and what we call osmosis, the moving of water across the membrane, and how the cell membrane controls what can come in and out of a cell in certain ways. And we're starting to learn that. So in water, you can see here, this is not saltwater, plain water, what the onion cell looks like versus salt water. I'm going to go back. So if we just look at the outside, okay, here's the cell wall. And then right inside the cell wall would be the cell membrane versus in salt water. What you're noticing here, it almost looks like it's shrinking back. And what's not shrinking back is the cell wall. That is still intact. What you're seeing here is the cell membrane almost starting to sort of shrivel up because of the content of salt, which is not good for the cells. So that much in the cell membrane, didn't have control of how much salt was coming across, but when you immersed it in a salt water, when it's not supposed to be in salt water, this is what's going to happen on a cellular level. And so this is a good indication of why, if you're really thirsty, like if you're stranded in an ocean, why you cannot drink salt water, that it will actually kill you, is because of what it's doing to the cell. So imagine this on a grander scale, like your body. It completely is going to break down those cells, and they're not going to be able to function normally. So if we look at this sort of together, onion cell and distilled water versus salt water. So here you can see you've got the cell wall and then the cell membrane and then the parts of the cell here. And you can see in this picture that when salt water is added, the cell membrane shrinks. You can see that the cell wall remains as it was and the membrane sort of starts to peel off. So that is not good because the cell membrane is supposed to, control what comes in and out of a cell. So tips for your microscope quiz and practicum is knowing the parts of the microscope. Make sure that you know what the body tube is and the eyepiece and the objectives and the stage. Knowing the functions of the parts and what they do. Knowing the total magnification of the three objectives, the red, the yellow, the blue, and basic things about the lab we did from this presentation. I might include some of the pictures on here. Do you know what happened to the cell membrane when put in when put in salt water, et cetera? So I'm going to have you just keep those microscope lab handouts for your reference. For the practicum, you got the rubric in class and you can just look over that. You don't have to memorize it, but just know that practice, practice, practice. In class, come in during study hall if you feel like you need extra practice or after school while you're waiting on your ride home. But I will give you the following directions. I will call you back to the microscope and I will say, place the slide on the stage and magnify to 40. So I would expect you to know how to do that. Then I'll look at your image. Is it clear? And then I will direct you to change the magnification to 100 and I will look at the image again. Then I will tell you to take the slide off the stage and leave the microscope in the proper magnification end position. And I'll be grading you on your ability to do that. Okay, thanks so much.